In this video, we'll begin the lecture on the peripheral nervous system. I'll discuss to a certain point in this lecture and then end it, and we'll continue this material in our next class period right before Thanksgiving. Keep in mind that as I discuss the peripheral nervous system, I'm going to be discussing material from chapters 13, 14, and 16, and it will encompass the structures that we do associate with the PNS, so sensory receptors, peripheral nerves and their peripheral ganglia, as well as motor endings of effector organs. We begin with receptors. Most of this information can be found in chapter 16. This will be the only section that we discuss from chapter 16 until we discuss the special senses in the very last lecture of the semester. So sensory receptors themselves, a receptor is any structure specialized to detect a stimulus, and that these are all responsible for responding to both internal and external stimuli in your environment. A sensory receptor is unique because it can undergo what is called transduction. And simple definition there is that transduction is the ability to transform energy from one form into another. Speaking in regard to nerve impulses, we say that transduction in a sensory receptor is when a sensory receptor begins to undergo some depolarization that then stimulates a postsynaptic potential of a sensory neuron. So the idea here is that we can liken these sensory receptors as the spark that ignites a flame, essentially. If a sensory receptor ignites a stimulus that can essentially be considered an EPSP, then that EPSP can then translate into an actual nerve impulse associated with an action potential. So the idea here being that a stimulus needs to be strong enough, and the sensitivity of a particular receptor can determine whether or not that stimuli will be enough to start the EPSP. Keep in mind that a stimulus is different than sensation. So sensation itself is the subjective awareness of the stimulus. Sensation is associated with the brain and the spinal cord, not with the receptor itself. And not all of these stimuli will make it to the brain. So showing you this image, you don't really need to memorize much from this image, but it's a nice schematic of where a receptor works in the process. If something touches your skin, for example right here, this little graph of the skin, you already know that regions of the somatosensory cortex are going to be responsible for feeling that. You also know that the somatosensory cortex is going to receive information from third order neurons, which receive information from second order neurons, which are then going to receive information from first order neurons. So by moving backwards here, you see that your receptors themselves are going to actually be the receptive endings of a first order neuron associated with an ascending path in the spinal cord. Here they're showing you what are called free nerve endings, muscle spindles, and what's called a joint kinesthetic receptor. With each one of these nerve endings, we see that they're going to be classified in a few different ways. And so we tend to classify sensory receptors on the four kinds of information that they can actually convey. The first is what's called modality. Modality is the type of stimulus or sensation that a sensory receptor is associated with. We also know they're capable of dictating location, intensity, as well as duration. So we'll begin by discussing the different modalities of sensory receptors. So again, a particular stimulus, modality, these receptors can be referred to as mechanoreceptors if they respond to a mechanical force like touch, vibration, pressure, and stretch. They're referred to as thermoreceptors if they respond to changes in temperature, photoreceptors if they respond to changes in light, chemoreceptors if they respond to changes in chemicals in either the air we breathe, the food that we eat, or the blood contents, the ions, the water that are circulating by specific chemoreceptors in the blood vessels. We also have what are called nociceptors, and those are going to respond to extreme stimuli that have the capability of producing pain. So this could be excessive force, excessive temperature change, so forth and so on. So again, that's how you group a receptor based on modality. Grouping your receptors based on location, you tend to use three terms to classify this. Exteroreceptors, detecting stimuli external to the body. Interoreceptors, detecting stimuli located on the internal viscera and blood vessels, and proprioceptors, a word we've used many times now, detecting changes in body position and movement. These are found in skeletal muscles, ligaments, tendons, joints, and all the connective tissues such as the fascia and the periosteum that cover our bones and muscles. Now, anytime you're looking at location, these neurons' receptors tend to have what's called a receptive field. So this is the particular region monitored by a single sensory neuron. In this illustration, we see first off what a receptive field is. So once again, here's a single sensory neuron. Here are its receptive nerve endings. And so from here to here is illustrated that this single neuron's receptive field. So this neuron is responsible for responding to any stimuli that occur within this receptive field. However, over here, you see that this particular region of the skin is going to be monitored by 3 separate neurons, each with a smaller receptive field than the one that you see right over here. So now you'll notice that one large receptive field like this cannot actually distinguish what we call two-point discrimination. And so because this particular neuron here is going to respond to stimuli in this region, period, it cannot distinguish the fact that there are 2 different needles right here that are trying to stimulate the skin. So it has very low two-point discrimination. Your back is a great example of where neurons have large receptive fields, so you'll find that you're a little less sensitive to two-point discrimination here on this part of the body. However, when you look at the fingertips or even the bottoms of the feet or your lips or your tongue, you find that the neurons tend to be more numerous and they tend to have smaller receptive fields. This increases their two-point discrimination. So very well illustrated here by the fact that this particular needle is going to stimulate neuron number 1, while this particular needle stimulates neuron number 3. So your brain then will become aware of the sensation of both. So we've talked modality and location. Now we move to signal intensity. So of course a weak stimulus versus an increased stimulus strength works like this. A weak stimulus is going to stimulate nerve fibers that have the greatest sensitivity. Recall this is associated with neurons that have really low thresholds. So it's very easy to depolarize towards an action potential. Now as stimulus intensity rises, you start to see that in addition to these greatly sensitive neurons, now you begin to stimulate less sensitive neurons, those that have a higher threshold. So you've got more neurons now that are firing. You also find that the frequency of nerve impulses is going to start to increase. So each one of these sensory neurons begins to fire more often, releasing more neurotransmitter at its synapse on down the line. And then you begin to see recruitment of nerve fibers taking place at a pretty quick pace. What's neat is the brain can actually start to interpret the combinations of frequency and recruitment to begin to determine sensation intensity from all these signals. Lastly, we look at receptors classified by duration. So duration of a stimulus is encoded by changes in the firing frequency within the passage of time. So in addition to knowing how intense a signal is, if the frequency continues to last for a long time, that's how you know when the stimulus starts and when it stops. Now, what's tricky here is that most receptors can undergo adaptation, where they can actually alter their sensitivity to a stimulus, even though a stimulus itself may continue to fire. So the types of adaptation we see are called phasic or tonic receptors. A phasic receptor tends to be what we call fast adapting. What that means is that it's very sensitive right off the bat, and it alerts the brain with a burst of impulses at the very onset, but then stops firing. So it quickly adapts to the stimulus. Oftentimes, these phasic receptors will send off a single final burst at the very end of the stimulus. And so you tend to see that the brain utilizes this to help you identify smell, hair movement, pressure. And these are important. If you are constantly aware of all the smells around you all the time, you'd probably go a little crazy. I think Febreze commercials refer to this kind of phasic reception as nose blindness. Tonic receptors then are a completely different story. These adapt more slowly, or sometimes they don't adapt at all, and so they tend to generate signals a little more steadily. Proprioceptor is the greatest example of this. You need to always be aware of whether or not you are standing, of whether you are holding something, or of whether you have uncomfortable joint position at any point in time, because these are all sensations you need to be able to act on at any given moment. Given your knowledge of the cerebellum and how it would respond to these proprioceptors, you can appreciate the fact that these proprioceptors never stop sending the cerebellum signals. So when we start to look at the structure of these receptors, we find that they can be referred to as non-encapsulated or encapsulated. I give you this table from a different textbook. I don't want you to memorize this table, but it does a nice job of showing what an encapsulated nerve ending or an unencapsulated nerve ending looks like. So these are free nerve endings right over here. You tend to find that these are associated with external reception in the skin, as well as internal and proprioception in the muscles and the blood vessels. But these nerve endings are just basically anchored in whatever tissue they're meant to monitor with nothing around them. It's a great example of non-encapsulated. The Merkel's discs that you learned about in the skin or the integumentary system, they are not wrapped with anything. So they're considered non-encapsulated, but they do end on the tactile discs or the tactile cells that we saw within the epidermis. Likewise, hair follicle receptors were just free nerve endings that wrap around a hair follicle, still considered non-encapsulated. Encapsulated nerve endings would include the tactile receptors and the Pacinian corpuscles that you learned about with the skin. Each one of these, the nerve receptive endings, are going to be wrapped up in a connective tissue bundle. And so that's what makes them encapsulated. We'll also see that muscle spindles and tendon organs, as well as joint kinesthetic receptors, are encapsulated as well. We will talk more about those when we begin to discuss reflexes later on. So before we can discuss cranial and spinal nerves, you need to make sure that what a nerve is very familiar to you. Let me begin by saying a nerve and a tract are very different. Tracts are going to be bundles of axons within the CNS, which is why we saw white matter tracts in the brain and white matter tracts in the spinal cord. Anytime you see the word nerve, that should immediately remind you that we are looking at structures in the peripheral nervous system. So the nerve itself is a cord-like organ, parallel bundles of peripheral axons enclosed by successive wrappings of connective tissues. So you see it right here, and this image starts to look familiar to a lot of people because this is the same kind of structure your skeletal muscles have. Let's start way down here. Here's unmyelinated and myelinated nerve fibers wrapped right here with what's called perineurium. Sounds a lot like paramecium. And so that gives you what would be fascicles of these individual axons. Bundles of fascicles tend to be laced together and surrounded by what's called epineurium right here. And the epineurium itself tends to be the outer cover of the nerve. So anytime you are studying a nerve itself, this is the picture I want you to get. I also want you to note the fact that it's highly vascular. So when you look at diseases such as diabetes mellitus tends to slow blood flow to particular large nerves, and that's one reason you can end up with nerve death. And that gives you myopathies and loss of sensation. It's one of the primary reasons that people with diabetes mellitus stop feeling the bottoms of their feet and can often end up with open wounds because they don't treat damage with care. So as we begin to discuss spinal nerves and cranial nerves in class, I want you to start picturing this structure. This structure is the main reason that we can discuss any kind of a spinal or cranial nerve as one that contains only sensory fibers, only motor fibers, or could be mixed because it contains neurons that are both sensory and motor. And that is where we will stop.